restriction enzymes were these things discovered in bacteria back in about the 70s, and they've grown to become these really useful tools in genetic engineering. What they can do is they can cut at specific points depending on the restriction enzyme being used. They cut at specific points when they see specific sequences of DNA. So what we're able to do is cut out and remove particular genes and insert them into the genes of other organisms to either try and replicate them or have them become part of the DNA and genetically engineer and change the particular characteristics of a particular organism. So if we have a look at these two sequences of DNA here, one is plasmid DNA, and the other one we're going to consider, we'll assume, is human DNA. So the plasmid DNA has come from a bacteria. So bacteria have these loops, these little loops of DNA outside of their large circular chromosome called plasmids. And these can be really beneficial to us in genetic engineering. We can insert genes into a plasmid, and bacteria will rapidly divide, and we get lots and lots of copies of a gene of interest. Our human DNA on the right here contains a particular gene that we want to try and replicate. So a restriction enzyme will cut at a certain location. They will cut, they're an endonuclease, which will cut when they see a certain repeating pattern of DNA. So the repeating pattern of DNA that for this example, our restriction enzyme is looking for is CCGG, like so. So many restriction enzymes cut in what we call a staggered fashion. So this restriction enzyme is going to cut at the start of the CCGG sequence on one strand and at the end of the GGCC sequence on the other. So if we use, let's just call this restriction enzyme 1X. So let's assume this restriction enzyme 1X cuts in this sort of fashion, this staggered fashion. Now, if we then look at the human DNA and we introduce this restriction enzyme to this human DNA segment, it's going to cut in exactly the same manner. The start of the Cs and at the end of the Cs on each of those strands. So once this has occurred, it creates something called sticky ends. So if I highlight the way that it's been cut, we're going to get this overhang. So we see our GGCC on each of those different segments now overhangs, and if we divide up or split up quickly TATGGCC, our two segments, they're now going to look something like this. So we can see there we've got these overhangs like so. So I refer to those as sticky ends. And I guess the reason why they're called sticky ends is because they are able to stick to other complementary sequences. So that's really helpful for us if we are trying to embed this human DNA into our plasmid in order to replicate a particular gene. So what we can see happen is if we take, actually I might highlight these just to make it clear. If we take our yellow segment of the plasmid DNA and we take our yellow segment over here of the human DNA, what we'll end up with is these two chopped up segments of GGCC, these two chopped up segments of DNA from the human and the plasmid are able to bind together in a complementary fashion because of those sticky ends. So what we'll end up with is in our plasmid we now have a bit of human DNA. So in order for these two segments to be joined together, we also need the help of another enzyme called DNA ligase. DNA ligase is going to assist in connecting those two segments together. So what we can see is restriction enzymes are able to cut at specific locations, cut plasmid DNA or human DNA in order to create these overhangs or these sticky ends. We can then use these sticky ends to join together something like human and bacterial plasmid DNA. We might be able to use this bacteria to replicate quickly and create more copies of a gene of interest by DNA ligase being able to connect or embed a human gene cut out into a plasmid, like I've demonstrated here. So restriction enzymes are really, really helpful in genetic engineering processes and to remove genes and create extra copies of genes of interest.